And now it's time to move on again and to go into real food production and processing. So the next speaker is Alexander Matis from ETH Zurich. Professor Alexander Matis is a food technologist. He is professor in sustainable food processing in Zurich and in Singapore. And he was also head of the bioeconomy department at German Institute of Food Technology, DIL Institute, and is an expert in non-thermal preservation and sterilization technology. So we are really, we can't wait to hear from you about what is on pipeline for production and processing. So the floor is yours.
Thank you very much, Chiara. Thank you very much for the invitation also for this very relevant panel. And I would like to thank the previous speakers, Takis, Berna, and Georgia, for bringing up really a perfect floor for the topic of emerging food production processing for more sustainable food. I want to give you some principles which we apply at ETH Zurich for developing emerging food production and processing, and I want to give you some examples regarding microalgae and insects on the following slides. So I would like... Okay, perfect. Yeah, I had to do 2 clicks. Okay, so first of all, why we need to think about emerging food production, I want to give you some arguments, and there are much more. We will go through a systematic approach a bit here, so you will see much more. But first of all, our food is produced on 14 million square kilometers arable land, and this arable land is completely used already. By the way, 1/3 of it is used by wheat, maize, and rice only, so by 3 crops. So that shows also the strong dependency on monocultures, but it shows also that if we want to produce more food on the left side, you can see the numbers, we need to produce more per square meter arable land. And we do that already with a very high efficient agricultural system, but also a massive use of fertilizers, which is using a massive amount of non-renewable energy for production of fertilizers. We can increase the arable land by cutting the rainforest, for example. I strongly recommend not to do that. Unfortunately, it's still done. And we can produce food or biomass on non-arable land surfaces. And here, I believe fermentation can contribute to our agricultural production. And I want to show you some examples about that. On the right side, you can see the increase of meat production. So protein-rich animal-based foods deliver a lot of important nutrition. However, they are linked to a lot of negative environmental impacts. And that's why we are looking for alternatives to this protein-rich animal-based foods. By the way, this is not only protein, which we are looking for. Animal-based protein-rich foods deliver a lot of important bioavailable micronutrients, and we will also discuss that a bit. On top, you can see the huge numbers of animals which we currently farm. So we farm more chicken than human on our planet, 19 billion chicken globally. We slaughter 69 billion chicken every year, an absolutely extreme number from my point of view. And these animals are fed with quite often food and, of course, feed-grade material. So we can also use that feed maybe for food production. Think about plant-based innovations to replace protein-rich animal-based foods. On top, we need to also feed our pets, 470 million dogs, 370 million pet cats, and they are treated quite often like little kids. And here we also feed a lot of protein-rich animal-based products to these animals. So there are significant challenges to think about alternative protein-rich foods. And I would like to focus a bit here in terms of food innovations on that category. What else is important? You can see here some recent publications on food system sustainability. So we need to go through a systematic approach, and that includes not only environmental sustainability, like CO2, land use, and water use, etc. It also includes the other 2 dimensions of sustainability, the environment, the economy, and the social side of sustainability, which includes nutrition and health. That is a bit connected in these reports, where I was really glad to join this Nature Sustainability Expert Panel led by Professor Chris Barrett from Cornell University, where I learned as a food engineer, the major impact comes from social innovations, such as behavioral change. But we can connect behavioral change with technology change, especially when we think about policy. And here also EFSA can be a great partner in supporting new policies for example, really leverage and support innovations. So of course, our current production can benefit from efficiency improvements, and that is done every day, I think, in our food production facilities. We can go through the technology, the behavioral change upwards to sufficiency. That, for example, that we focus on policies that induce behavioral change, for example, avoiding long-distance air travel for vacation or other purposes. We can grow the technology change direction from efficiency to substitution, that we, for example, develop renewable energy technologies, which give us then the source of energy which we need also for our food production to have lower CO2 footprints. Or we go then the middle, sufficiency and substitution in both ways, in strong support with policy, for example, and then really get to a more impactful outcome. And I think we need to combine these two angles much more. Okay, what else is important? From the technology side, I'm a food engineer, so we go through a reverse engineering approach. You can see here on the left, affordable nutrition as number one KPI, at least for our purposes in research and development. The second is organoleptic properties, which are needed, for example, such as taste, to convince consumers. And we need to understand also the consumer better, what they want and what they like and what they don't like. And the third, I'm sorry, that is a little typing error, would be the improved three-dimensional sustainability performance, which I already introduced briefly to you. That is generated by structure in nano, micro, meso, and macro scale. If we really think about that from the molecular structure to the macro scale structure, and here we can use then materials with techno-functionalities, such as, for example, texture in the mouth, or also biofunctionalities, which can, for example, lead to positive health impacts, which is then also regulated and evaluated by the EFSA. And that needs, for example, the process which is generating the structure. And here, food process engineering can contribute to this reverse engineering approach, where we really want to then deliver affordable nutrition with focused organoleptic properties such as taste and more sustainable indicators and performance. However, technology development needs to go through the innovation cycle, and we can see here the technology readiness level evolution. If we have a great idea in our research facility, for example, in our laboratory in academia, where we got a lot of support from the taxpayer, we develop it further from a lab scale to a pilot scale, hopefully to a nice food pilot plant. But then there is a drop of resources around technology readiness level 4 and 5. Very often projects in academia are then somehow finished. Why we should go on higher than 100 liter per hour or 100 kilogram per hour scale. Industry, our partners are saying, well, this is not efficient enough. We can't put it into a food factory. So please continue the journey to industrial scale feasibility. And here small businesses, small, medium-sized enterprises, and specifically now more and more spin-offs and startups from our environment can support this. And one nice example is Planted, a ETH spin-off, which now collected 70 million francs to develop plant-based food innovations in their new factory in Kemptal. So really, they could scale it up. And now the big international industries is also very much interested on these innovations to take it up into their facilities and to industrial scale up to, let's say, 10,000 kilograms per hour capacity. If we scaled it up, like the most modern insect factory some years ago in Protex in the Netherlands, that was a very nice feasibility of that technology. However, we need 1,000 of insect factories for feed application, for example, to replace the 5 million tons of fish meal globally, to have really here a more sustainable feed application than also available for consumers and industry. So the economy of scale, so multiplying then this highly optimized principle is also needed on top, and only then we can have also a competitive price structure to be really also viable on our market, to be also comparable to benchmark products which are highly optimized and highly scaled, and also already with the economy of scale. Okay, what else is important? I think when we launch, for example, new biomass, and microalgae will be an example I want to discuss with you a bit more, we need to think a bit the biomass value pyramid. So I'm not so much in favor using microalgae lipids, which are having even a health claim accepted by EFSA with the polyunsaturated fatty acids, to use that directly as bioenergy. That is a waste of resources. So let's use that as maybe functional food ingredients. And then maybe let's think about some bulk ingredients which we can extract. And last but not least, let's extract material for bioenergy. There is always, from my point of view, the priority: first material use and then second energetic use. And that is a bit integrated into our approach at ETH Zurich and the Singapore ETH Center, where we want to support single cell value chains, which are shown here on the top, specifically focused on microalgae, and also alternative animal-based value chains focused on insects grown on site and waste streams to deliver emerging feed applications. On the bottom, you can say that we have a data group which is only analyzing all the time our sustainability performance of our research project results and assure that we really go to a more sustainable direction. You can quantify sustainability, and we will see some examples of these methodologies. In this slide, where we go now a bit from this bottom approach to our food system sustainability methodology. Again, a multi-indicator sustainability analysis is needed here to have a fair comparison of new products on the market with a benchmark, not only environmental sustainability. For example, life cycle assessment was mentioned, and especially for food application, LCA, life cycle assessment, has a lot of limitations because it's quite often mass-based. It doesn't account the nutritional quality, potential health impacts of the new food products, because that is social sustainability. And you can see here then also the third dimension, economy. We suggested here the 25 sustainability indicator-based methodology based on leveraged in 7 domains: food nutrient adequacy, ecosystem stability, and then already we enter into the economic side, affordability, availability. Do we have enough volume available for our global food production? Are they affordable? We need to have affordable nutrition. Nutrition shouldn't be a luxury. Social cultural well-being includes animal welfare, resilience, mainly relevant also and was very visible during COVID. For example, nations like Singapore then initiated new big policies to triple their food production to increase their self-supply rate because the food trade was impacted. Food safety, I don't need to talk too much about that. I think in our developed world, quite a great luxury which we have and is given very often. Consumers don't think too much about it, but in developing areas, we need to improve the food safety. Waste and loss reduction, I think we really need to do better, specifically with the food waste which we generate. That is all linked to the 17 Sustainable Development Goals, where food systems are the heart of these SDGs. So there is a great opportunity for us to really contribute here more. One more clear example maybe of this very high-level methodology, you can see here the combination of life cycle assessment with a nutritional dimension, the Nutrient Rich Food Index, NRF called, where we quantified and summarized 21 beneficial nutrients and 2 disqualifying nutrients on the y-axis, and then the mass-based environmental impacts on the x-axis. We used here regional specific data sets from different regions, where we have, for example, also different data sets already available now. And you can see variations in different regions regarding environmental sustainability and nutritional quality. And that is needed, for example, to have a fair comparison. And this is based, for example, for a kilogram of product in different food categories and so on. So these methodologies are available. LCA is even ISO normed. And let's use that to really quantify sustainability performance and to avoid too subjective conclusions regarding sustainability performance. Yeah, and now we enter into some solution space, so insect-based site and waste stream utilization for alternative feeds. We believe we can here contribute to alternative animal-based feed applications such as fish meal when it's not produced in the EU. Quite often it's based on small catch, and this is a significant biodiversity loss in our ocean. So what can we do? We can first of all use site and waste streams if we consider 2 important aspects first. First of all, we shouldn't produce any waste or side stream. So we should prevent it. We should reuse it or reduce it. And if that is not possible anymore, then we can use these substrates, for example, for insects as a feed substrate to produce alternative feeds. And this is done in our insect colony in Dübendorf, close to Zurich, with the Aquatic Research Center of ETH Zurich, and also in the Singapore ETH Center now in a highly optimized way, which I will introduce to you on the next slide. Where we take the side streams and waste streams, depending on the region. In the EU, only side streams are allowed, no waste streams, but globally, a lot of waste streams are also used, and I will give you one example for that. The waste and side streams are pre-processed, and then we add the black strutterfly larvae, which is a kind of extremophilic species from the insect group, which is really delivering in a very fast way in liquid and also pasty waste streams, high fat containing, high protein containing larvae. So up to 30% fat, 30% protein is feasible. In dry matter, you have even more. And that can be then delivered as insect-based poultry, aquaculture, or pig feed. And that is, for example, also regulated already in EU if it's grown on side streams. But this is a biotech approach which can also deliver feed applications in decentralized and centralized ways in developing areas, from my point of view, if you have the know-how how to operate such a black soda larvae colony. Here are shown here, you can see the products, the protein meals, the insect lipids, and the residues can be used as functional fertilizers. Here we collaborated also with industry. The industry uses side streams, but we, again, in developing areas, are also considering waste streams here and evaluate the safety of these production chains. One example to use waste streams is here in Singapore, the Singapore ETH Center with our project coordination at the National University Singapore, Professor Nalini. And you can see us here in the colony where we go through a high-tech approach with sensor-based technology to detect physical parameters, behavioral parameters of the insect larvae, which are fed them with heterogeneous food waste, which you can't avoid, which you can't reduce. That should be the highest priorities first. But if that is not possible, then it's delivered to the larvae. As it's a decentralized production, we need to have highly automation in this decentralized production. We generate data sets which are then delivered to a deep reinforcement learning model, and which is then generating the best conditions for the larvae to have the highest performance as feed application. We can then also take these data sets, making it open access for low-tech applications, because these data sets can also predict performance if you use heterogeneous site and waste streams as substrate for larvae with their varying conditions. Another example for an alternative value chain can be a microalgae-based value chain. There are 200,000 species known. We use very few of them only, and some of them are also currently under the novel food evaluation. For example, a very exciting microalgae called Galdiera superraria, which is grown in volcano craters. It can be grown on pH 1, so no contamination because of the acidic conditions, and it can deliver very important, relevant macro and micronutrient concentrations, which are currently evaluated for bioaccessibility and bioavailability in our laboratory. We also work a bit on the production via our bioreactors of that specific microalgae, which can be grown in an oxygen and CO2 balanced mixotrophic cultivation. I will briefly touch that also later on. We develop pulse electric field systems to better stimulate and extract ingredients. We developed process technologies to structure, to preserve these new biomasses, and also collaborate here with nutrition, medical science, and consumer science. And I want to introduce some of these research to you. You can see here that you can grow microalgae in different ways. You can see our wave reactor here in a little movie. You can grow microalgae with photoautotrophic cultivation, which is then delivering green microalgae. However, here we have a limited concentration, let's say maximum 20 grams per liter, only possible because you need to integrate the light into the biomass. And the opposite is the heterotrophic cultivation. Here, the carbon source wouldn't be CO2. It would be, for example, coming from side streams. It would be carbohydrates. The energy source would be also carbohydrates. And you can also grow them in the middle with mixotrophic means, where you give a bit light and also a bit carbon for energetic and carbon needs. With that, you can change the pigments, chlorophyll, carotenoids. You can change the nutritional profile and also the final color. Microalgae is not a new topic. Microalgae cultivation is known since thousands of years. The Aztecs consumed already microalgae. We discuss it since decades also for emerging food production, but it wasn't really flying, maybe because of the limited concentration, the high energy inputs, and also limited viable products on the market. And viability comes a bit from color. The dark green color was not so attractive for consumers, one aspect. We could now increase the concentration before harvesting up to 100 grams per liter with heterotrophic and mixotrophic cultivation. We can now grow extremophilic microalgae. At pH 1, we can reduce technology demand, and we can also therefore reduce capex and the total cost per production. And that is maybe then leading to certain opportunities. Sometimes Photoautotrophic cultivation is the only way, and I'm not sure whether EFSA is also considering these applications. Who is taking care for European astronauts, for example, when we produce food in space? I don't know which agency would be then responsible for the food safety, but we did already a feasibility trial at the International Space Station, growing chlorella vulgaris, producing oxygen for the cabin crew, and also food biomass as a kind of feasibility in a long-term cultivation in a 600 milliliter reactor. You can see here the German astronaut Alexander Gerst growing the chlorella. We got it back with a Soyuz capsule before the war, luckily, and could analyze the biomass then regarding microbial ecosystem and microbial flora, and detect, by the way, also safety relevant microorganisms, because in the autotrophic cultivation, normally you have also other microbes. So we would need to have a food safety concept also for such applications in life support systems, and that is more and more discussed because there is a Moon and Mars station maybe planned later on. But this is really then the long-term scope for food safety regulations. Let's go back to Earth, where we developed a nanosecond pulse electric field technology to again increase efficiency of the production chain to make microalgae-based foods more affordable. Currently, there are some interesting products, but the limitation is definitely the too high price of such products because the primary production is not efficient enough. At ETH, we developed this cell stimulation technology based on a new approach where we use, for example, less than 500 joule per kilogram only, which should come, of course, from renewable energy sources. And we could demonstrate for several single cells that they can grow faster with the given resources and the given time in comparison to the control group. So we obtain even up to 40% more biomass using nanosecond pulse electric field cell-based cell stimulation. That is not completely understood how that works physiologically, but we could obtain more biomass, which is then leading to, again, less price per kilogram biomass for final food applications. So that can really contribute to more affordable food production. And that was then also commercialized. You can see here my first doctoral student, which started with me in 2017 at the ETH Zurich. He finished in less than 3 years, developed 4 patents, 7 peer review articles, and could win the ETH medal for his great doctoral research. And then went to industry and commercialized this technology from his doctoral research into an industrial prototype, which we now installed the first time worldwide in the Singapore ETH Center for cell stimulation application, not only for microalgae, also for yeast, for bacteria, and maybe also for stem cell application. Yeah, let's see the outcome of that. However, we talked a lot about innovations, and I think it's a very nice example that research can be also implemented fast enough if there is a strong connection to industrial partners which are willing then to take over and also take over the risk, of course, of such a new technology development. Yeah, another example I want to show you here more on the downstream domain of single cells, but again with a case study on microalgae for structure process property interactions and emerging process technologies. For single cells, which are grown in a quite high concentration of water, we maybe need to rethink our approach. We shouldn't apply maybe the plant-based approach, growing biomass and then, let's say, kill the organism, harvest it, and then process it. Maybe we can milk cells in suspension and running this cultivation in a continuous mode. And this is possible over months. Here again, microalgae, which can be grown in acidic conditions, for example, at pH 1, can be beneficial because in a long-term cultivation, contamination is always this challenge. Currently, we can grow Galdiera over weeks and months, for example, and applying pulse electric field, in this case, microsecond pulse electric field, would enable to extract free proteins, not every protein in the cell, but some proteins, and we keep the cell alive. You can see here 2 graphs or 2 functions where we have the extraction and recovery. Of course, we can extract more protein, but then the cell would die off. That is done in a classical up and downstream approach in serial ways, but maybe we can do up and downstream together in a parallel approach, milking proteins out of cells and then separating that with, for example, ultrafiltration in a bypass, in a fermenter, which maybe need to have a different approach than our plant-based production. Yeah, if we then extracted protein fractions, the best is, of course, if you have an early value chain or an innovative value chain to add or to leverage functionality. Functionality justifies a higher price on the market. That can be techno-functionality, but it can be also biofunctionality. Here we can see examples of relevant techno-functionalities for food production. For example, surface activity. That we have here protein fractions where we investigate them with interfacial biology, demonstrating that we can emulsify with these fractions different phases like water and lipids or fat phases, and then generate interesting new emulsions, replacing, for example, animal-based emulsifiers like whey protein applications. And then it depends a bit how fine your emulsion needs to be. On the right side, you see a highly purified protein isolate, which takes a lot of effort to produce, with very, very thin interfacial layers. On the right side, on the left side, you can see the extract, the crude extract from the Atmosphera platensis, also known as spirulina. Maybe this more rough, let's say, emulsion is also okay for your food application. You can reduce resource investments and, of course, also have a lower price of the production per kilogram. And maybe this emulsion is still okay. So it depends a bit on your final application. But that is a nice demonstration of the techno-functionality. And we observed a lot of interesting techno-functionalities from protein fractions from microalgae. Yeah, if we then go more for the bulk protein, and that is the major discussion about novel protein, again, I would like to refer more to protein-rich foods as an alternative. We can combine microalgae with plant-based protein concentrates. Here in the feasibility study, the first time worldwide, with bright yellow microalgae grown in heterotrophic mode, so in the dark. And here we have Oxana-chlorella protocoides, which is not novel food. That is already double-checked. And we could demonstrate that we can produce meat substitutes. Here, in this case, up to 30% microalgae integration. In our current running R&D, we could demonstrate already up to 80% microalgae integration, still keeping the structural formation, which is then very comparable to chicken breast, for example. So the feasibility could show, okay, that is already possible. However, price-wise, it would be quite challenging to put that into a market. But what I also like, we could demonstrate the increase of nutritional quality. Because I think many alternative protein-rich foods to the animal-based origin, they don't have the nutritional equivalence. So we, in the end, suggest an alternative, but not with the same nutritional quality. And here we need to work, food scientists, food technologists need to deliver similar nutritional quality if we want to replace animal-based foods. Otherwise, it would be an unfair comparison also. Here we could show with the vitamin E, B1, B2, B3, and B6 concentrations we could increase by adding microalgae. We could increase these concentrations, and even after a quite intense extrusion process, these concentrations were still much higher than the soy-only controlled product. Yeah, what else we can do? We can also, of course, investigate the nutritional quality, and that is currently our main focus on our R&D, where we also collaborated with Nestle, where we could demonstrate, for example, feasibility studies for microalgae lipids. You can see here the application of pulse electric field processing on Chlorella vulgaris, increasing the bioaccessibility of lipids from microalgae, which are very relevant for nutrition. Because very often maybe they are in the product, but they are not bioaccessible because there is a cell wall around. So using pulse electric field in combination with incubation, we could increase bioaccessibility, but still keeping the oxidative stability of the lipids, because if they are, of course, delivered directly to oxygen, they would oxidize and the product would be not okay for consumption. Yeah, I hope I could show you a bit our approach on emerging food process technology development based on emerging sources, also with the case study from the single cell domain on microalgae, where we believe this can deliver the most diverse nutritional quality in comparison to the other single cells. An alternative animal-based production chain based on black soldier larvae, site and waste stream utilization, and always aligned with multi-indicator sustainability assessment to really double check that we go to a more sustainable production. For the microalgae, we are currently trying to develop that into a biorefinery approach, where we have a cascade-wise extraction of sensitive ingredients and then more stable ingredients afterwards, based on many, many studies, which you can see here on the right. All of them are open access. We want to do that in an urban context, because we don't have arable land available anymore, we can go for non-arable land application. That is also currently done in our Singapore ETH center, where I will move now with my family in 2 weeks. And of course, if you are interested, please pass by for a little tour maybe. You can see here our translation project with industry partners from the startup side, but also from the multinational side, which support us for using liquid side streams from the Singaporean food industry in an heterotrophic microalgae cultivation, where we use our emerging pulse-electric field process technologies to stimulate, but also extract ingredients. We go through a structure, process, property, interaction approach, where we want to structurize via 3D printing and extrusion products, which are then delivering meat substitutes and fish analogues. And here, which is also very important, we also ask Singaporeans whether they really want that already now. And the results so far show a lot of openness. By the way, here also the Singapore Food Agency is involved, which then also support us to deliver the safe but also affordable, hopefully nutritious food products in this approach. With that team, thanks very much to that team, and thanks to our partners from academia, applied R&D centers and industry. Thanks to the Sustainable Food Processing team in Zurich. They did all the work. I was happy to present it, and thank you very much for your attention.